Our second speaker in the area of kind of macromolecular quality control is Micha Rapé from the University of California, Berkeley and HHMI. Professor Rapé's lab has made many important discoveries pertaining to mechanisms that maintain cellular homeostasis, usually involving the small signaling protein ubiquitin. And today he's going to tell us about quality control pathways that protect cells from reductive stress. With that, I'll give the floor to Micha Rapé.
Let me start by thanking Roz and all the other organizers for, first of all, putting together such an exciting symposium. It's always really fun to mingle between disciplines. I don't get that luxury too often. So the only point, of course, is that I'm sad that I can't be there in person. But thank you so much for inviting me. So the story that I want to talk about today really started, pretty exactly 11 years and 9 months ago when our first daughter announced herself. The scientist that I am, I was getting very nervous because I had read all these papers about pediatric diseases, and they appear with a probability of 1 in 1,000, 1 in 5,000, 1 in 10,000. If you read enough papers of that sort, you can get a little bit anxious. But then I had to calm down, and I figured out that actually when you look around, development is quite the opposite. It's extremely robust, and most babies that make it look like babies, and trust me, behave like babies. And that robust development really happens even if the conditions can be quite distinct. So I, for example, was born in an area of Germany that was called Bavarian Siberia at the time. One reason was the god-awful weather. The other reason was that it was really plagued by rampant environmental pollution. And so despite the toxins in the air, despite the toxins in the water, my mother gave birth to something that some would agree looked like a human and behaved like a human. So development is really robust, even though it can be very difficult. And key to that robustness of development are stress response pathways. These are signaling pathways in organisms that have two arms: a sensor arm that can detect a stress, such as a lack of nutrition, a lack of oxygen, chemicals, and so forth. And then an effector arm that, in response to detecting the stress, sets into motion basically reactions that can take care of that situation. These stress response pathways have initially been investigated most heavily in the protein misfolding field, where loss of stress responses cause protein aggregation and often neurodegeneration. And that led to a view that they are particularly important during aging. But when people then knocked out components of stress pathways, they realized that they are actually very important very early on in tissue formation and early development. And the role of stress pathways in tissue formation is still very poorly understood, and that became sort of one of the key questions we were asking in my lab. And we realized early on that this is actually a two-way street. So by studying stress responses, you can understand why development is so robust. But on the flip side, by studying development, you really have a playground to discover new stress response paradigms. And so the story I want to talk about today is a recent example from the lab where we studied a particular developmental pathway to find a new and very important stress response system that provides quality control of the electron transport chain function. Now, I have to say that all of this work was done by an amazing team of scientists, and most notably, it's the brainchild of a postdoc in the lab, Andrew Manford, who has been the leader and is still the leader of this project, really conceived it and put it forward in an amazing way. And he was helped by many in the lab and outside of our lab, but most importantly by Elijah Mina, especially for the second part of the talk, where we look at the atomic details of the stress response pathway. So I owe great thanks to these two amazing scientists. Now, when Andrew came, he was thinking about which developmental paradigm to look at, and we pretty quickly narrowed down on myogenesis for several reasons. First of all, it's a pathway where the core components are understood very well, and so you can focus on regulation. It is also a pathway that you can reconstitute in vitro, and that makes it easy for us to screen for new players in the system. And then most importantly, it's a developmental system that's known to be regulated by stress. So for example, if you work out in a gym, or during the pandemic, if you run around in a park, you impose exercise-dependent stress on the system. And hopefully, if you do it right, that will induce this differentiation program and you get a little bit more muscle. So in order to find new stress components, Andrew focused on a particular class of regulators called E3 ligases. These are proteins that modify other proteins with a polyubiquitin chain that can then be recognized by the 26S proteasome for degradation. And we focused on these E3 ligases because practically every single stress pathway that had been discovered so far has a core key E3 ligase as one of its components. So Andrew took out E3 ligases in myoblasts and asked whether that had effects on myogenesis, and he found a really intriguing one called KEAP1, that when he depleted it, completely eliminated muscle formation or myotube formation, as shown by the absence of these green myotubes. And that sort of immediately validated our approach because KEAP1 is a stress response gene. It's the master regulator of the oxidative stress response, which means that you're having too many reactive oxygen species that can damage proteins, DNA, or lipids. If these arise, they inhibit KEAP1 by means that are still very poorly understood. This leads to stabilization of the NRF2 transcription factor, which then is the effector arm that puts into motion antioxidant signaling pathways that can deal with the stress. Now, when you have oxidative stress, that's transient, and so once you take care of it, it goes away. But when you deal with this genetically, as we did in the screen, you persistently activate NRF2 and antioxidant signaling, and we thought that this might actually be the cause for the problems in myogenesis, that you might titrate away reactive oxygen species that have signaling roles that might be deleterious. And that's something you can test very easily. If this problem in differentiation is because you stabilize this transcription factor for too long, you should rescue this by co-depleting NRF2, and that's shown here. You can indeed fully rescue myogenesis. You can even do this by taking out antioxidant targets of NRF2, such as this enzyme that's important for glutathione production. And we could take this one step further. If this really indicates that it's the absence of reactive oxygen species at their physiological level, which inhibited myogenesis, then you should do this chemically as well. And so we can take scavengers that siphon away reactive oxygen species from their major sites of production. These are enzymes of the electron transport chain in mitochondria. And if you do this, you don't have reactive oxygen species and you cannot differentiate into a myotube. And so this is a condition where we lack reactive oxygen species that is described as reductive stress. It's sort of the less known sibling of the oxidative stress response. It's actually a really important stress condition. It was shown in 1987, around 1987, that reactive oxygen species have signaling functions, and these are essential for development. So if you experience this stress, this likely has very important consequences for development, as seen in our differentiation approach. It is a condition that occurs very frequently. For example, if you eat badly and glycolysis doesn't run full steam, that can lead to side products of glycolysis that can inhibit KEAP1 and produce reductive stress. But also KEAP1 is very frequently mutated. It's one of the 3 most frequently mutated tumor suppressors across all cancers, and it can be inhibited by protein aggregation, for example. So this might actually occur quite often. Yeah, we don't run around with cancer or other diseases that can arise upon unmitigated reductive stress. And so it was clear that there must be signaling systems in our cells that can detect the absence of reactive oxygen species and set in motion, an alleviating response. So there has to be a reductive stress response, the components of which were not known. And so we were lucky because we had a genetic system to find them. Basically, the hypothesis was that if we would co-deplete components of a reductive stress response with this KEAP1, with our initial condition, that should again rescue myogenesis, as you had seen it in the examples I showed before when we co-depleted NRF2. When we did this again, focusing on E3 ligases, we got one really stunning hit, an E3 ligase called FEM1B, which shown here completely rescued myogenesis in this screening approach. So FEM1B is part of a larger E3 ligase that has a scaffolding composed of colon-2, an active site composed here, which transfers ubiquitin. And then FEM1B basically defines substrate specificity. Through experiments I don't have time to show you, basically trapping mass spectrometry, we identified a protein called FNP1 as its essential substrate that was polyubiquitinated by this enzyme and sent for degradation by the 26S proteasome. So if this indeed is a component of a reductive stress response, there's a very clear prediction, and that would be that this degradation event occurs only when you lack reactive oxygen species or when you experience reductive stress. In order to monitor this, we needed a quantitative assay to see that, measure degradation. And for this, we established this system where in 2 years of very hard work, we defined a peptide of about 20 residues in the substrate that was both sufficient and required for binding to the E3 ligase. That's the so-called degron. We fuse this degron to GFP and then express it together with mCherry that's driven by an internal ribosome entry site. And so the ratio of GFP to mCherry basically is determined by how well this FNP1 degron can drive degradation. And so normally you get GFP to mCherry ratio is shown here, and the green curve is our control. If you now induce the E3 ligase, you see that the curve shifts to the left, and that means it's now degraded. And that requires activity, because if we have a substrate binding ligase or a catalytic inactive ligase, we don't see this. And we also know this is really specific because we can take out the E3 ligase, in this case using CRISPR, and now we see that the curve shifts to the right, so you have more GFP indicating that this is stabilized. And so having this very quantitative assay now allowed us to measure whether the levels of ROS affect stability in this case. And for this, we wanted to do it in the most physiologically meaningful manner. And so most ROS in cells are produced by mitochondria, especially complexes 1 and 3 of the electron transport chain. In the cells that we used, they basically got fueled by glutamine, which is taken up by mitochondria via glutamate, and then entering the TCA cycle that produces substrate for the electron transport chain. So if you would starve cells of glutamine, the electron transport chain would run out of substrate and you would lack ROS and you would impose reductive stress. But you can also feed cells a downstream product, alpha-ketoglutarate, which basically means that the TCA cycle would run in hyperdrive and now you would produce more ROS. You can ask whether that affects degradation. This is the result, and green again is the control curve. In red is glutamine starvation, where we deplete reactive oxygen species, and you see the curve shifts to the left, meaning accelerated protein degradation, just in the same way as inhibition of KEAP1 actually accomplished. And in blue is addition of alpha-ketoglutarate, meaning more reactive oxygen species, which shifts the curve to the right, meaning now we have protein stability. So this is a system that indeed reacts to reductive stress by inducing degradation of this FNP1 protein. So we have the core components of the sensory arm of the reductive stress response, which measures the presence or the absence of a physiologically important reactive oxygen species. And very briefly, of course, you need an effector arm to counteract this stress. Most reactive oxygen species are produced by mitochondria, so naturally we looked at those organelles. And I'll just show you one little result, because this is published, which is the most dramatic result that you can see by transmission electron microscopy. So normally mitochondria are these little beans. If you get rid of the substrate in cells that don't experience stress at the time, nothing much changes. But if you deplete the ligase and you would stabilize the substrate, you see really drastic changes on mitochondria. They first of all turn black. That's a phenotype that was first described in the 60s as matrix condensation, and it means that these mitochondria run out of substrate for the electron transport chain. Many mitochondria also overproduce cristae to yield these onion-shaped structures. This is a response to an inactive electron transport chain. The cell tries to overproduce enzymes of the electron transport chain to compensate for the lower catalytic activity. And then in many cases, we also see initiation of mitophagy, which is just another indication that these mitochondria were inactive. And most importantly, all of these phenotypes were completely rescued if we co-depleted just the single substrate FNP1, which, together with all our other experiments, showed that FNP1 degradation actually leads to activation of mitochondria, and as such, activation of the organelle that produces most of the reactive oxygen species in the cell, and so counteracts the stress. Now, this was sort of our discovery of the stress response, which raised, of course, many questions. And two of them are really the ones that I really want to highlight very briefly today. One is that we need to know how this is regulated. How can the cell detect the absence of reactive oxygen species? And then, of course, is that actually connected to development? So in order to understand regulation, we focused on this degron that I mentioned before, and it was very conspicuous very early on that it contained 3 highly conserved residues, the only fully conserved residues, and all of these are cysteines. And that, of course, given that we're looking at some redox reactions, suggested that oxidation might actually control this reaction. And so we could test this directly. We made this peptide with a fluorophore and then could monitor binding to the ligase by fluorescence polarization. When you do this well in the presence of a reducing agent, you see a very high affinity binding. If you take this peptide out just for a few seconds in the air, it is oxidized. We know that these cysteines form intramolecular disulfide bonds, and we actually know that these cysteines are oxidized in cells that don't experience stress in intramolecular disulfide bonds. And under these conditions, you don't see any binding. Now you take these samples, you add back reducing agents, so you resolve these disulfide bonds, and you get, again, high affinity interactions, showing you that reversible oxidation controls access to the ligase. So how does this happen on a molecular level? This would be the first ligase that's controlled by reversible oxidation of a degron. And so how is the enzyme able to read out the oxidation state? For this, Andrew and now Elijah solved the crystal structure of the FEM1B FNP1 complex. And it revealed something really interesting. The degron accesses a deep pocket in FEM1B, but quite strikingly, it doesn't bind any amino acids of FEM1B. There is no side chain interactions or peptide bone interactions, which normally always mediate protein-protein interactions. By contrast, what it does, it sandwiches two zinc atoms that act as a molecular glue to tether the substrate to the ligase. And that's better seen here when we zoom into this from a structure. There's really no interactions between the side chains, but sorry, my light is out in this energy efficient building. But what happens is that cysteine residues, these are the three conserved cysteine residues of the degron and the histidine residue of the substrate. And then histidine and cysteine residues of the enzyme really just sandwich this zinc. So zinc is a Velcro that stitches the substrate to the enzyme. And these cysteine residues can only chelate zinc when they are reduced. They cannot do this when they are oxidized, giving you an immediate explanation why this is a redox-sensitive system. So it turns out that the cellular sensor for physiological ROS is an E3 ligase that depends on zinc as a molecular glue so that it can only recognize the reduced form of the substrate, but not the oxidized form that would be present in the absence of stress. And so the second question, of course, was, is that important for development? And we already early on, figured that this might be because there is a particular mutation in the ligase that causes a syndromic developmental delay. And that mutation, it's an arginine residue, is very interesting in its position. It is in the substrate binding pocket, but it doesn't touch the substrate, and it doesn't promote binding affinity to the substrate. In fact, in our structure, it coordinates a buffer molecule through a hydrogen bond. So when, however, we placed this mutation into our quantitative assay for looking at degradation, we saw something very interesting. Here we induce the ligase. We see again that the curve shifts to the left, that it is meaning that the substrate is degraded. But if we put in the disease mutants, they shift further to the left, showing that these are hyperactive mutations. It's an enzyme that causes a mutation that causes a toxic gain of function. And with all the experiments we had done, we could predict that this is due to the fact that most likely this amino acid is important in recruiting an inhibitor of the reductive stress response. So for reasons of time, I spare you how we discovered it. It's a family of proteins called the so-called BEX proteins. I just show you the data for this. In green, again, is the degradation reaction that we normally have. We induce the ligase in red, it shifts to the left. Then we induce the ligase in red together with this inhibitory protein, and it goes back to the right, meaning it really prevents degradation. If we do this now with a disease mutant, the first thing that you see is, first of all, the curve shifts further to the left, meaning, again, it's a hyperactive mutant. And now if you co-express the inhibitor, it's completely resistant to it. It cannot move back to the right. And what is striking about this is that these genes, the BEX genes, they are all encoded on the X chromosome right next to each other. They are deleted in a syndrome called XQ22 deletion syndrome, which removes just these BEX proteins. And it turns out that the phenotypes of this disease are practically identical to the phenotypes of mutations in the reductive stress ligase, meaning that mutating the binding site for the inhibitor or eliminating the inhibitor interferes with development. And that really shows you very nicely that accurate reductive stress signaling or accurate monitoring whether the electron transport chain is active is actually essential for development. So with this, just to sum up, I'm sorry, this is the reductive stress response that I think validates our approach that development is an amazing playground for discovering new stress response pathways. It is a system that monitors the electron transport chain. If the electron transport chain is inhibited, you don't produce reactive oxygen species. That leads to reduction of these cysteine residues in the degron, allowing you to form this very unique enzyme substrate pair, leading to degradation of NIP1, activation of mitochondria, and production of ROS, and that is very tightly controlled during development through these pseudo-substrate inhibitors, as we call them, of the BEX family. There are a few bigger picture issues that we are very excited about, I just want to mention one of them, and that is that this is now the reductive stress response. It looks very similar in its architecture to the oxidative stress response, as well as to the hypoxic stress response. And now we have three basically stress responses that monitor the redox state of a cell. They all look very similar. They're all built around cullen-based E3 ligases. They're all regulated by reversible oxidation reactions. And they're all targeting exactly the reaction that you would need to target in order to counteract the stress. And I think this is a really neat example of convergent evolution, where you highlight that there is a family of enzymes that is perfect for the regulation that you need under these conditions. And of course, this work opens up a ton of new questions for us. Most importantly, we try to understand how the reductive stress response is integrated into larger metabolic systems and networks. For this, we do lots of genetic interaction screens right now. We try to understand the physiology, because I think this really shows a function of ROS as second messengers in monitoring electron transport chain activity. And we can now ask, how important is that for development? For this, we've just generated knockout mice in FEM1B, and we're in the process of generating the R126 mutant knock-in mice. And then finally, we are actually drug discoverers in my lab. We try to translate what we find into new approaches for drug discovery. And so we have begun to very actively see whether we can find small molecules that interfere with the signaling system, providing us with a small molecule handle to either turn on or turn off ROS production, dependent on the therapeutic need. And so with this, again, thank you so much for the invitation. It's really a big honor to be here. Most importantly, again, this is really work driven, spearheaded, and done by Andrew Manford with really important help from Elijah Mina. And of course, I'm also very grateful for our many collaborators, particularly John Kurian, who has been extremely generous in teaching us X-ray crystallography, which we didn't know before, and of course, the funding sources. Thank you.